Hi, I'm AMENA Smith-Fine, and I'm a neurologist working in the Moser Center for Leukodystrophies at Kennedy Krieger Institute. Today, I'll give you an overview of what we've learned from a natural history study of the disease LBSL, which is a leukodystrophy that features progressive ataxia with onset usually in childhood. The approach we've used to study LBSL can be applied to related rare diseases that affect motor development, including Syngap1. An exciting aspect of this project is that we're using wearable sensors to get information about walking and balance during teleresearch visits at patients' homes. And this is especially relevant in an era where the ability to conduct research remotely is essential. So leukoencephalopathy with brainstem and spinal cord involvement and lactate elevation, or LBSL, is an autosomal recessive disorder caused by mutations in DARS2. This encodes an enzyme that's critical for translation of respiratory chain complex proteins. The MRI pattern in LBSL is very distinctive and features white matter changes in the cerebrum, cerebellum, and brainstem and spinal cord tracts. And then there's also usually an increased lactate peak on MR spectroscopy. Clinically, patients experience progressive ataxia, spasticity, and dorsal column dysfunction, and the onset is usually in infancy or childhood, although occasionally not until adulthood. Earlier age of onset has been associated with earlier loss of independent walking and wheelchair dependence. Of course, these are the outcomes that we would hope to intervene on and avoid in this disorder. So our objective is to establish clinically meaningful quantitative outcome measures for clinical trials in LBSL. Our approach is to obtain information about gait and balance using wearable sensors and an established clinical ataxia rating scale. We perform assessments during virtual visits at participants' homes, as I mentioned earlier, and this is to increase participation and decrease the burden of travel for these families. We are also doing testing in age and sex matched healthy volunteers. Today I'll be showing baseline data, but we repeat the assessments every 6 months over 5 years. So we chose the Opal system of accelerometers as this is a non-invasive, relatively inexpensive technology for recording movement in a real-world context. The system has been used to measure disease-specific characteristics and other neurodiseases, including MS, Parkinson's disease, and cerebral palsy. And similar metrics of gait and sway have been reported in other ataxic disorders. The Opal system consists of 3 watch-size battery-based accelerometers that submit a signal to a central sensor through low-power radio frequency waves. And the sensors are just placed on the feet and the waist for a brief series of tests. So for the virtual visits at home, we send an Opal's kit, a web camera that's remote-controlled, a laptop, and an Opal device manual that's in simple layperson language to explain the tests and the equipment setup. This is just a picture of one of our participating patients and family. As I mentioned, we also used as a measure the scale for assessment and rating of ataxia, or the SARA. So this is a brief and sensitive tool for clinically rating ataxia. It's been used in SCA and Friedreich's ataxia previously and has shown excellent test, retest, and inter-rater reliability in these disorders. The SARA was able to show progression in SCA after a year, but it did not capture all ataxia symptoms reliably early in the disease. However, wearable accelerometer measurement of gait ataxia was shown to be superior to the SARA in the early stages of SCA. So we're exploring the use of both of these measures to assess disease severity and progression in LBSL. In terms of our clinical characteristics of our cohort in LBSL, this is based on a sample of 8 patients. Our median age was 10, with a range from 5 to 23 years of age. Basically, we had a minority of patients that were female. The mean age of symptom onset was about 5 years of age, and generally patients always presented with ataxia and fine motor delay. 3/4 of patients experience an episodic deterioration in the disorder, meaning if there was a fever, a viral illness, a cold, they had a temporary regression in motor symptoms, but then a later recovery after a week or so. And then in terms of overall gross motor function as measured by the GMFCS, the scores were low. So on a scale from 1 to 5, with 1 being mild and 5 being the most severe impairment, most patients only had a rating of 1 or 2, and then the manual ability classification scale, or the MACS, also were in the mild range of 1 to 2. The SARA ataxia scale is rated from 0 to 40, mild to severe ataxia, and you can see that the mean was only 8, so patients were mildly affected, mild to moderate. Okay, so this is a video of a young woman, 10 years old, with LBSL performing a walk test. So this is a 22-foot walkway. Participants perform 2 trials at a time, and information about the gait cycle is collected. So first of all, what we found in terms of the stride length during the walking test is that the stride length, and again, the stride length is the forward distance traveled by the foot during a gait cycle. This is the first initial contact to the next initial contact of the same foot. And a short stride length is a compensation for unsteady gait. So as hypothesized for patients with ataxia, our LBSL group had a shorter stride length compared to their age-matched controls. This difference was not statistically significant in our small group of participants. And then next, it appeared the time that it took to complete a gait cycle was slightly longer for the LBSL cohort. And finally, the LBSL group had a slower stride velocity compared to the control group. And per the literature, a difference of this degree of about 0.1 meters per second is actually clinically meaningful, although we did not see a statistically significant difference. The hallmark of ataxia, of course, is variability, which can be estimated by the lateral step variability. So when considering 3 consecutive foot placements made by the left or right foot, the LSV is defined as the variability of perpendicular deviations of the middle foot placement from the line connecting the first and third. And this basically indicates a veering or stumbling gait, and larger deviations might also indicate a wider base to preserve stability during the walking. So our LBSL group showed a greater lateral step variability versus controls by about 2 centimeters, which was statistically significant. Next, we examined circumduction and elevation at mid-swing. And so this is basically the distance that the foot travels outward during forward swing. So we saw that the LBSL cohort exhibited a slightly greater circumduction value compared to controls. And in terms of the elevation at the mid-swing, there was also a statistically significant increase in this value in LBSL patients. We next considered turning during the walk trials and found that the LBSL participants took slightly longer to turn with a smaller turn angle, and that resulted in a slower turn velocity in degrees per second versus controls. So together, these findings indicate balance and stability during the turns for these patients. Next, to move on to talking about standing balance, our remote balance testing consisted of 4 postural sway conditions in which we challenged participants by variably removing visual input and or narrowing the base of support. So 4 conditions. They performed 2 trials of each condition, and their stance width is standardized with a foot plate. Sway is measured from the lumbar or waist sensor, and there are 10 key variables that are collected by the sensors in 3 different planes.
10 seconds left.
So to demonstrate,
left.
These clips represent LBSL patient participant performance on the balance test. Above are plots mapping this girl's sway path compared to an age-matched healthy volunteer. And basically this is looking for them top down. So you can see that across all conditions, there is a greater sway for the LBSL participant, but especially in conditions when her eyes are closed, and it's greatest in conditions when her eyes are closed, in the condition where her eyes are closed, also with a narrow base of support. And I'll just demonstrate again each of these.
Almost done. Less than 10 seconds left. 10 seconds left.
Okay, so here are the sway plots for the control and LBSL groups, and below we've quantified the sway area for the groups. So as in our representative patient, we just saw the sway path is greatest and the feet apart, eyes closed, and the feet together, eyes closed conditions. And then to further evaluate the impact of removing visual input on sway, a Romberg quotient was calculated for each participant. This is the sway area with the eyes closed divided by the sway area with the eyes open. And you can see that the average Romberg quotient was significantly increased for LBSL subjects, which likely reflects proprioceptive contributions of the dorsal column medial lemniscus pathway fibers in the spinal cord. So to us, this meant that the myelopathy that's seen in LBSL may be an important contributor to their overall motor disability. Lastly, these plots are demonstrating that the SARA ataxia score is strongly correlated with the lateral step variability during walking in the first plot and the sway area during standing. This supports the idea that the wearables are capturing variables that are clinically meaningful and do represent ataxia. So in summary, through virtual testing at home, we found that LBSL participants walk and turn slower, have shorter stride lengths, and show more gait variability compared with healthy volunteers. The step variability and sway area measured by the wearables is strongly correlated with the clinical ataxia scale, the SARA, which we think is likely related to sensory or cerebellar ataxia. And then finally, removing visual input has a greater impact on the sway area than narrowing the stand space, suggesting that the myelopathy or the dorsal column dysfunction is really an important contributor to instability while standing. And so again, this is demonstrating the power of these wearables in a specific leukodystrophy, but this is an approach we do believe can also be applied easily to related rare diseases affecting motor development, such as Syngap1. All right, and thanks to everyone who participated in this research. Certainly, of course, number one is our LBSL participants and families, and who also gave permission to show the recordings today. Ali Fatimi is the principal investigator and my mentor who has worked with this project, worked with me on this project, as well as Amy Bastian, who directs our motion analysis lab at Kennedy Krieger. Thanks to Constance Smith-Hicks, who gave the invitation to participate in the Young Investigators Research Day, and to the rest of our amazing team. Thank you very much, and I'll take questions live.